So looking back, I left off on Thursday last week talking about oxidation reduction reactions. Okay, oxidation, we learned, is the loss of electrons from a molecule, and that is going to also typically involve the loss of hydrogen atoms. Whereas in the case of reduction, this is the acceptance or the gaining of electrons during a redox reaction. Okay, those two reactions are coupled to one another. In fact, what that just means is that anytime one substance is oxidized, another one is simultaneously reduced. Okay, so that's why we call them oxidation reduction reactions or redox reactions for short. They go together. Okay, we're going to look and spend the majority of our time today talking about the oxidation of glucose, which is where we're going to take glucose and we're going to strip it of a lot, not all of its electrons, and we're going to strip its hydrogens to the point what's left over is carbon dioxide. And that's important because when you oxidize something, typically that's exergonic or energy releasing, and that therefore can be utilized to generate ATP. Okay, the vehicles that are going to carry those electrons and those hydrogens are NADH plus H plus, or NADH2 if you want to shorthand it, and FADH2, which in that case they described as reduced coenzymes because they've gained those electrons. Okay, when they give those electrons and those hydrogens up, which they do to the what we call electron transport chain, which is what you're actually seeing here in this illustration, those processes, and it's not shown here in this illustration, are eventually used to transfer that energy to generate ATP. And that's the whole goal of cellular respiration. Okay, so like I said, we're going to be talking mainly about the oxidation of glucose. Okay, so I just want to kind of introduce carbohydrate metabolism before we start getting into it. Now, before any of that oxidation of glucose can take place, and this relates back to what we talked about last week, we have to get that glucose from our intestinal lumen inside the small intestine into our body. Because remember, if something's like in your stomach and your small or your large intestines, it's technically not inside of us. Okay, and so before glucose and other monosaccharides can be used in the body, they must be absorbed from the small intestinal lumen via a process known as sodium cotransport, which is what's illustrated here in the bottom left figure, where essentially, and we talked about this, is what we talked about last Thursday. This would be the inside of your small intestinal lumen. These, this area would be the inside of those absorptive cells of the small intestine. Basically, sodium, since it's going to go from this area to this area down its concentration gradient, it gives glucose a piggyback ride. And that's why we call it sodium cotransport. And then that brings the glucose truly into the body for the first time, into our own cells. Okay, and then eventually the glucose will make its way into our blood circulation. And once in that circulation, the fate of that glucose is dependent upon the needs of our cells. What we'll spend the most of our majority of our time here for this several next slides is the use of that glucose to make ATP. But we can also use that glucose to do other things. Okay, if we don't need to synthesize ATP, because we might already have a whole bunch of glucose circulating around, we can actually convert it and form amino acids. We can store it in the form of glycogen, and that process is called glycogenesis, which if you think back to our endocrine lecture, that's one of those 6 buzzwords that I wrote out for you guys and told you guys that you would need to know. Glycogenesis ends with genesis. That means it is a formation or creation. In this case, glycogen is at the beginning of the word, so it's glycogen genesis, if you want to think of it that way, the formation of glycogen. And then we can also even convert glucose into triglycerides, which are forms of fat, in a process called lipogenesis, the synthesis of lipids, or in this case, the type of lipid, a triglyceride. Okay. Now, like I said, we're going to spend the majority of our time talking about the production of ATP, okay, which means that we're going to have to bring that glucose into our body cells, whether this is a muscle cell whether this is up in our brain, whether it's down in the tips of our toes. Okay, what we utilize to get that glucose from the interstitial space or extracellular fluid into the cytosol is through what we call glucose transporters, or GLUTs for short, GLU for glucose, T for transporter. These are a special type of carrier or transporter specific to carrying only glucose, and it's a transporter, so it physically changes its shape. It's not a channel like what we've seen in many other cases. But once inside the cell, typically what happens to that glucose is it's attached to or a phosphate's attached to it, I should say, and that's because of kinase enzymes forming a glucose 6-phosphate, or as what it's listed right here, phosphorylated glucose, which at this point, once it's got that phosphate on there, it's kind of trapped, which then allows the cell to hold on to that glucose and get it ready for this. And that is cell respiration. Now, if we think back to our lecture on the respiratory system, just a couple lectures ago, we used the word respiration a lot. We said that there's 5 basic steps of respiration, and that included pulmonary ventilation or breathing. And then we talked about pulmonary gas exchange, alveolar gas exchange, where oxygen first enters the blood. Then we talked about the opposite of that, which is systemic gas exchange, where oxygen leaves the blood to go into cells. And we also talked about, in that same lecture, the transport of oxygen as well as carbon dioxide. I'm kind of ignoring carbon dioxide, but that's also exchanged and it's also transported. The 5th process of respiration is this, and I told you guys at that point we would talk about it later on. Well, here we are at that point. Okay, this is the process that releases energy from food to produce ATP. In other words, this is known as the oxidation of glucose. Once again, the stripping of its electrons in order to produce ATP. Okay, now ATP is a form of energy, but it's not the only form of energy that is produced as a result of this process. Another form, and we talked about this the other day, we've also talked about this before, just like any other chemical reaction, this chemical reaction is going to result in the formation of heat. And the benefit of this is that heat that we produce is not just wasteful energy, it gives us the body temperature. Okay, but the real purpose of this reaction is to produce ATP. Okay, and we can get a lot of ATP, not quite as much as what you can get in like fats, as we'll find out later in the lecture, from glucose, because there's quite a bit of potential energy in it. When it's broken down to carbon dioxide and water, this energy is released in the form of ATP as well as that heat. Now, the ingredients for this include not only glucose from our diet, but also the oxygen that we have talked about that comes from breathing. The CO2 that's formed is the same CO2 that we talked about that we must exhale to breathe out of our body so that we don't become acidic internally. Okay. The water that's formed is pretty insignificant. We call it metabolic water because it's formed during metabolism, but it's not enough water that we could like literally just stop drinking, because that, we don't produce enough of this. Okay. So it does help meet our daily need, but it's not sufficient enough for us to survive. Okay. And as I said, the energy in the form of heat gives us body temperature, but the true reason and why we do all this, and the most important aspect of it, is the ATP that's formed, which can be then used for any energy requiring reaction. And think about all the different things we've talked about this semester that require energy, whether it be muscle contractions, which we learned about that, not too long ago, where it changes the position of the myosin head so that it can flex and pull on the thin filaments. Talked about, pumping stuff against their concentration gradient from low to high, like in the case of like the sodium potassium pump. Other examples of just like moving stuff around inside our cells, moving chromosomes during mitosis. So ATP is needed for just so many different things, and this is how we produce it. And now the synthesis of ATP means that the energy is used to bond a free phosphate, which is just a singular phosphate, to a molecule of ADP, which is adenosine diphosphate, which means that there's some stuff, but just kind of the most important thing is the diphosphate, which means there's 2. And if you put those 2 phosphates and a third together, you got ATP, which is adenosine triphosphate. And then that gives the cells their energy. And as a result of this use of energy, what we form is ADP free phosphates. Essentially, that third phosphate is removed. And so these things are very present in our cells anytime we have utilized energy requiring processes. Now, the breakdown of glucose summarized above is not quite that simple, however. It involves a complex series of equations where glucose is taken apart piece by piece, with also the removal of hydrogens and the splitting of carbon-to-carbon bonds. This releases energy of glucose gradually so that a significant portion, around 40%, is available to synthesize ATP. The other 60 or so percent is going to be the heat that's produced. So it's quite a bit more complex. And if you want a little bit of a better detail on how complex it is, and we're going to go through these individually here for the next several slides, there's 4 major sets of reactions, or kind of 4 major steps. The first of which is glycolysis. This is a 10-step series of reactions, which results in the formation of a little bit of ATP, a couple of those electron-carrying coenzymes, okay, electron and hydrogen-carrying, I should say, coenzymes, NADH plus H plus. But we also form what's called pyruvic acid. Now, notice where this is all taking place. It's not inside of our powerhouse, not inside our mitochondria, but instead in the cytosol. And not only is it occurring out there, but this is the only step out of the 4 that is anaerobic. And what does that mean? We've seen these words aerobic, anaerobic before. What does anaerobic mean? Remember? It means it does not require oxygen. Okay. The other 3 steps, which include step 2, the formation of acetyl-CoA, or as I typically call it, the transition reaction, that requires oxygen. So does the Krebs cycle, which is step 3. And then finally, and this is the major way that we produce ATP, is through the electron transport chain. We only produce a little bit of energy in glycolysis. We don't form any in the transition reaction. We only form a couple during the Krebs cycle, but what we form a lot of are those electron carrying and hydrogen carrying reduced coenzymes that carry those electrons and those hydrogens to the electron transport chain. Okay? And this is where not only do we form the ATP, this is actually where we'll convert the oxygen to water. There's the metabolic water that we talked about, and so on and so forth. Then we'll get into all this. So I didn't read through that, and that's okay, because we'll start talking about it right now. We'll begin with the first step, glycolysis, which literally means splitting sugars or breaking sugars. That's another one of those of our 6 big buzzwords. Lysis is a fancy way to mean breaking down. And that's what we start to do in this process. I wish it was called glucolysis. That would make it a lot simpler, but it's glycolysis, which is the splitting of a 6-carbon glucose into 2 pyruvic acid molecules. Now, you'll see in this lecture, probably, as well as maybe even in the video, I can't remember how they word it, but sometimes you'll see pyruvic acid listed as pyruvate. And this is not chemistry class, and so I'm going to consider those things as being the same. They're truly not, but for our, all intents and purposes, it is. Okay. Now, in order to split that glucose into those 2 molecules, it actually requires a little bit of energy. Okay, a couple molecules of ATP are necessary to start this process, which is why we call this step the energy investment step. Okay, and this energy that's required is the energy of activation, which makes the glucose molecule unstable enough to break it down. Okay. And so we go from a 6-carbon glucose molecule, and we form half of that, which is 2 3-carbon molecules. Okay, and then those 3-carbon molecules are going to donate some high energy electrons to the NAD plus, forming NADH plus H plus, which are then going to be carrying those electrons and hydrogens to the electron transport chain. We do make a little bit of ATP though, about a net, or not about, it is a total of 4 ATP. Okay, and this is a form of substrate level phosphorylation. But as you'll see on the next page, and I'll just jump there, in parentheses, behind 4 ATP, it says a net of 2, and I'll explain why that is in just a second. This, once again, is occurring out in the cytoplasm, not inside of the mitochondria, and is an anaerobic process, meaning it does not require oxygen. Okay. Now, looking at this illustration, it's a fairly simplistic view of what's taking place, kind of broken down into two phases or steps, the energy investment phase and the energy harvest phase, which is significantly simpler than the 10 steps that it really is, which is illustrated on the next page. This is the figure that your textbook actually provides for you. And it really, to me, just doesn't give any information to a student that's capable of understanding it, at least in, at your level of schooling, unless you've taken lots and lots of chemistry classes. I'm sure this just looks like a bunch of letters and random numbers. Okay. I like this image for that reason. Okay. This is actually taken from an old biology textbook I used to use when I taught biology. But what we have, and we'll just start here on the left with our glucose molecule, are 6 gray dots. And these gray dots are representing the number of carbons that are in that molecule. Remember, glucose is C6H12O6. C6 tells you that there's 6 carbons. Okay. What we do immediately, and it's during that energy investment phase, is we use a couple ATPs and we transfer a phosphate from each of them and we stick them on the ends. And in doing so, that basically stresses the molecule to the point where it breaks in half. And so what we are left with are 2 3-carbon molecules and a phosphate attached. Okay. We will then attach another phosphate so that there's one on each side. And that's going to pay off here in a second. But between that point, we oxidize it, which, remember, oxidation means that we are snagging electrons, we're pulling electrons, and we can see those are shown right there, and hydrogens, okay, forming NADH plus H plus. It doesn't show it, but remember there's that other hydrogen that's there, it's just not directly attached. So if you really wanted to, you could write in this, this little blank here, plus H plus, same thing down here, plus H plus. Okay. So we oxidize it, snag some electrons and hydrogens. Those phosphates that are attached get transferred over to some ADPs, and that forms 2 ATP here, 2 ATP here, for a sum total of 4. But why do I say it's a net of 2? Okay. Well, I always just think of it this way. I'm sure some of you have never been to a casino because some of you are probably not of age yet, but I'm sure everybody knows how a slot machine works. Okay, slot machine is a pretty simple device. And so you put in a couple quarters, you pull the lever, boom, 4 quarters come out. Okay, if you put 2 quarters in and 4 quarters come out, how much money did you win? 2 quarters. Okay, you don't win $1, you don't win 4 quarters because you started with 2 quarters. Okay. Now I'm trying to explain this to my wife. She thinks, I guess, the glass is fully full, because if she walks into the casino with $20 and walks out with 5, she won $5. But exactly. But I guess it's just that she sees it as entertainment. Not that we go to the casino very often. But my point still remains. It's truly, you're only gaining a net of 2 ATP. Okay. Which, if we think about it, what we've learned this semester is a total net of 2 ATP is not going to provide much energy at all. Okay. What we're left with as a result of this process are 2 of these pyruvic acids, or as I said, you'll even see it listed as pyruvate, which is, for our class, equivalent to pyruvic acid. Now, if that walking through it doesn't make sense, let's watch a little video that will hopefully bring it home.
How do you turn that vital food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that vital food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule. releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistants. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen...
Okay, I'm cutting it off right there. What she was getting ready to say, I'll get into in just a second, and then we're going to finish watching the video as we kind of roll through. I'll start and stop it a couple more times. But for what you need to know for the exam, I'm not going to ask you to walk yourself through all of those 10 steps. I'm not going to ask you to know, exactly specifically what's taking place throughout the reaction. Okay, stuff that you're definitely going to want to make sure you know where it takes place. In this case, in the case of glycolysis, it's the cytoplasm. Know that it's anaerobic, meaning not requiring oxygen. And you're going to see this for all of the 4 reactions. Make sure you know what the inputs and the outputs are. What you start with, what you end with. Those are the things that I would ask you to know for the exam. Now, what she was getting ready to say is what happens next with those pyruvic acids. She was going to say when oxygen is present, what takes place is the pyruvic acids get pulled into the mitochondria for further aerobic respiration processes. And I call that aerobic respiration processes because this is the aerobic pathway. Okay? And that means that it requires oxygen, where we bring the pyruvic acid into the mitochondria. We prepare it to enter the Krebs cycle by forming what's called acetyl-CoA. But that's not always possible. In some cases, what has to take place, because oxygen is not plentiful, is an anaerobic pathway. Okay? When oxygen is scarce, what we do is we convert pyruvic acid to lactic acid. Okay, and we're going to talk about that on the next slide. Okay, but that's not the preferred mechanism. The preferred mechanism, and that's what we'll jump into after we cover this quick anaerobic pathway, is the aerobic pathway. We've talked about forming lactic acid before. Referring back to our muscle system lecture, we learned that lactic acid, and I talked about it being kind of like the ugly stepdaughter or the stepsister that gets blamed for a lot of things when it truly is not responsible for a lot of those things. People seem to think that it causes cramps and muscle soreness and joint stiffness and stuff like that. It has nothing to do with any of those things. It can affect your pH because it is an acid, so it can make your body become more acidic. It can also contribute to fatigue, and that is stuff that's actually been shown to happen as a result of lactic acid. So let's talk about this process where we form it, what's called fermentation. And like I said, this is not what we prefer to do, but it is something that we do if no oxygen present in our cells. Once again, what we'd call anaerobic conditions. And this happens, especially in examples like when you're very strenuously exercising, when your muscles aren't receiving a sufficient amount of oxygen, they become anaerobic. Okay? And if we're going to continue to exercise, we still need to continue to produce ATP. Okay? And in doing this process, what we do is convert pyruvic acid to lactic acid, and we call this lactate fermentation. Humans aren't the only thing that go through fermentation. Okay, yeast, for example, goes through fermentation. What do people use yeast for? Bread, making bread, right? What does it do for your bread? Makes it rise. When yeast undergoes this fermentation, it releases gases, and that's what makes your bread rise. Okay, fermentation happens in the formation of alcohol. Okay, and I'm not going to get into that, but that's not something that's unique to humans. Okay, as I mentioned, buildup of lactic acid in muscles can contribute to muscle fatigue. So what happens? Well, I've got a pretty great image to illustrate what's taking place in this process, which is actually kind of a two-part image. You might not recognize it, but I'm going to kind of explain what I mean by that. If I were to draw a line just right here, down the side of this picture from here back to here, this is just a simplified image of this image. It's just showing you glycolysis, where we start with a 6-carbon glucose and we end up with 2 pyruvic acid molecules. And in the steps in between, we form a net of 2 ATP, and we oxidize glucose and form some NADH plus H pluses, or 2 NADHs are just shown here. If I were to draw a line from here on the left side of the pyruvic acids to over here, this is all fermentation. Okay. What we do is we take that pyruvic acid and we reduce it. Okay. Back in glycolysis, we oxidized glucose, which means we took electrons from it. Now we're going to give those electrons and those hydrogens right back. And we can see that really nicely here in this illustration on the right. The hydrogens from the NADH plus H plus are given back, and they're attached right there. And that's how we go from pyruvic acid to lactic acid. Okay. And that's what we do. Now, does this, and can you see any ATP formed here? No. Okay. But what it does is it frees up our NAD pluses. It opens them up to the point where they can then go find a new glucose and oxidize it. And that's important because you can't form any ATP from a glucose without first oxidizing it. So by producing NAD plus, that helps keep glycolysis going. And that's the main takeaway, and that's what I would highlight, underline, whatever. We reduce pyruvic acid, forming lactic acid. It's not the lactic acid that's beneficial. It's forming the NAD plus so that we can find a new glucose and then start going through glycolysis there. Now, once again, remember during glycolysis, this is not a super efficient process where we form a whole bunch of ATP, but it is a process that does form ATP, so it does benefit us, but it's just not like a long-term solution. And it's not the preferred solution. The preferred solution is to go through the aerobic pathway, which is going to take place inside the mitochondria, and it results in the formation of acetyl-CoA, and then that eventually enters the Krebs cycle. And that step between glycolysis and the Krebs cycle is called the transition reaction. And so this is our second of our 4 steps. If I go back to this picture and this title or this information over here, it says formation of acetyl-CoA. And it's shown that way here in the figure. I like to call it the transition reaction, transition step, because it truly is. It's a step between glycolysis and a kind of much more important step, Krebs cycle. And it's not super crazy what's happening during this process. And a lot of figures, and your textbook and the video actually is going to do the same thing when I pick back up with it. They just lump this in as part of the Krebs cycle. To me, it's completely separate, but I can see why they lump it all together. And once again, this is only going to be able to happen if oxygen is plentiful, what we call aerobic conditions, which is going to involve the conversion of pyruvic acid to acetyl-CoA, which is important because acetyl-CoA or acetyl coenzyme A, as its longer version is what can actually enter the Krebs cycle. Okay. Now, this is going to take place inside of the mitochondria. If you don't remember mitochondria, and there was a pretty good picture of it here, has two membranes, an outer membrane and an inner membrane. But that inner membrane has these inner foldings called cristae or cristae. That's not where this is taking place. It's taking place in what's called the mitochondrial matrix, which is the fluid on the inside of that inner membrane. Okay? The cristae is where the electron transport chain takes place, as we'll talk here in a little bit. Okay? So let's find out what happens. Okay? There's essentially three processes that take place, and you can see 1, 2, and 3 listed here, where we go from a pyruvic acid to acetyl-CoA. Okay? First thing we do is we decarboxylate that pyruvic acid. It's just a fancy way to say we remove carbon dioxide from it. And we can see there's one of those CO2s. And this is the CO2, not the only CO2, but that's some of the CO2 that we breathe off. And that's where it's coming from. Now, carbon dioxide has one carbon, and there's that carbon, and that's why we could see 3 gray dots here. But here, acetic acid only has 2. It's only got 2 carbons at this point. Then we oxidize it by taking some of its electrons and hydrogens. And then we slap a coenzyme A to it. And in doing so, that forms acetyl-CoA. But if you look at my outputs that I've got listed here, we only show 1 acetyl-CoA, but I have 2 listed. We only show 1 NADH plus H plus, but I have 2 listed. I only show 1 CO2, but I've got 2 listed. Why is that? Because if I go back to this figure at the end of glycolysis, we start with the glucose, but we don't just end up with 1 pyruvic acid, we end up with 2. So if I really wasn't doing a little bit of a better job, I guess I could have just put this picture on the page twice, because this is only showing you 1 of those pyruvic acids. We have 2 that are formed at the end of glycolysis. Does that make sense? Okay. Do we form any ATP? No. So this is not an energy yielding phase. The next step, though, is it's just not super energy yielding. In this phase or step, whatever you want to call it, is known as the citric acid cycle, Krebs cycle, TCA cycle. I usually call it the Krebs cycle. And you'll see why we call it a cycle here on the next page when you have an illustration. Okay. And instead of reading through all this, I'm going to just basically jump ahead. But it is an aerobic process occurring in the same place that the transition reaction did, which is the matrix of the mitochondria. And it's a series of reactions in which a molecule of acetyl-CoA is taken apart and its carbons are converted to carbon dioxide. Okay. Here's what it looks like. This is a figure from your textbook. It's not the best image, but it is a decent one to show us what's happening. Okay. It's one of those examples that I mentioned where they've actually included the transition reaction as part of it. Okay. If I were to kind of fix this image, I would just kind of keep this part up here out. Because if I were to circle all this right here, this is that transition reaction. Okay. Which means the rest of this is actually the Krebs. So it involves acetyl-CoA, okay, which is a 2-carbon molecule with a coenzyme A attached to it. Right away, the CoA just gets removed, and that 2-carbon leftover acetic acid, essentially, gets added to a 4-carbon molecule that's not shown here, but it will be right here, and that forms a 6-carbon molecule known as citric acid. So that's where the name citric acid cycle comes from. Okay. And then what we end up doing with it is pretty significant. We oxidize it several times. We snag a bunch of electrons and hydrogens and form several NADH plus H pluses. For each rotation around, and that's once again why we call it a cycle, we form 1, 2, 3 NADH plus H plus and 1 FADH2. We remove a couple carbon dioxides, so we form some CO2. There's some more of that CO2 that we breathe off. And then we form 1 ATP. But notice here at my outputs, it's just like what we talked about with the transition reaction. There's 2 CO2s, but I've got 4 listed. 1 ATP shown, but I've got 2. 1, 2, 3 NADH plus H plus is shown, but I've got 6 listed. 1 FADH2, but I've got 2 listed. And that's because, same thing, we don't just have 1 pyruvic acid. We therefore don't just have 1 acetyl-CoA. How many do we have? 2. And so for every glucose that we start with, this process takes place twice. All right, let's pick back up with our video. So I'm gonna back it up just a touch. She's gonna end glycolysis and move right into the transition reaction.
Of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA.
Okay, so this is her running through the transition reaction. She kind of just does it real quick, okay? There's our carbon dioxides. There's our NADH plus H pluses carrying the electrons. There's our 2 acetyl-CoAs.
Process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called the cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand.
Okay, so if you didn't pick up on it, her math, what she said is that as a result of this process, we form 8 NADH plus H pluses, or 8 NADH2s, I believe is how she said it. She also said 6 CO2. Well, here my list is 4 CO2, 6 NADH plus H plus. So why is mine listed differently? Once again, that's because she's essentially just adding the transition reaction and the Krebs cycle together. So she's kind of just considering them the same thing, part of one another. So the 2 NADH plus H plus is here, added together with the 6. That's where the A came from, and the 6 CO2, 2 here, 4 from Krebs cycle. Make sense? Now, we've accomplished 3 out of our 4 steps. But at this point, have we formed much ATP? The answer to that is no, not at all. We formed a net of 2 during glycolysis. We formed none during this reaction, transition reaction, and we only formed 2 during the Krebs cycle. What we formed a lot more of are these NADH plus H pluses and a few FADH2s. Okay, going back to glycolysis, we form 2 NADH plus H pluses. Okay, when it comes to the transition, we form 2 more. So that's 2 plus 2, 4. And then you add 6 more to that, that's 10 NADH plus H pluses. And then 2 FADH2s, that's 12 different things that are carrying electrons and hydrogens. And this is where the true energy is going to come in and pay off here in the electron transport chain. The electron transport chain is a series of electron carrying proteins. So it's literally a chain of proteins that carry electrons or transport them, hence the name, containing copper, iron, and are found embedded in the inner mitochondrial membrane, which is that folded structure called mitochondrial cristae. Okay. The pairs of hydrogens that were once part of the glucose molecule are brought to these proteins by the carrier molecules of NADH plus H plus and FADH2. Each hydrogen is then split into a proton, H plus, and its electron, which is just abbreviated E with the negative symbol there because it's negatively charged. Electrons are passed from one protein to the next, and then finally oxygen accepts it. Okay, the reactions of these electrons with the proteins release most of the energy contained in that glucose molecule. Each oxygen that is gained, 2 electrons reacts with 2 of the hydrogen ions, and that forms water to H2O. So that's where the H2O water comes from. Okay. Once again, this contributes to intracellular fluid and also prevents acidosis, because if hydrogen ions accumulated, they would rapidly lower the pH of the cell. Here's what it looks like. And another example of a figure I've kind of actually taken from a different textbook to show what's taking place. We're going to bring those 10 NADH plus H pluses from the 3 different steps that we started, glycolysis, transition, and the Krebs cycle. And what we're going to do is we're going to donate the electrons to the very first of these proteins that make up the electron transport chain. And as you can see, that electron is just going to get channeled from protein to protein. Eventually, oxygen accepts it. And while that takes place, what we're going to do is we're going to also move hydrogens into what's called the intermembrane space, which is the space between the 2 membranes. Remember, inter means what? Between, right? Okay. And so a bunch of hydrogens are going to accumulate in this space, which creates a gradient. Okay. And eventually, as those hydrogens come down their gradient through a enzyme, ATP synthase, the energy that's produced there is converted into ATP. And it's a very significant amount. Okay. From this step alone, it's a total of 32 to 34 ATP. So from this step, we produce a heck of a lot more ATP than we did previously. Okay. And this is an example of oxidative phosphorylation. And we call it that because it's oxygen requiring. Phosphorylation is the fact that it's attaching a phosphate group to something else, in this case ADP, adenosine diphosphate, forming adenosine triphosphate. Now, why does it require oxygen? Because that oxygen right here is what eventually is going to accept those electrons plus the hydrogens that were brought there, and that's what forms water, which doesn't really provide us with much, which is why I listed it here as a byproduct. It does contribute to our intracellular fluid, but it's not a significant contribution. Okay, so let's finish the video.
Now, the majority of the energy is produced by aerobic respiration. We need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecule.
Okay, so what you guys can see is these little white dots are the electrons, these little plus dots, those are the hydrogen ions, the H pluses, okay? Remember, the NADH plus H pluses and the FADH2s, that's what they're carrying, electrons and hydrogens. And let me back it up. Now, if you see all these fancy names, be happy that you don't need to know what their names really are, okay? These are just the proteins that make up the electron transport chain.
Carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred.
Look what's happening. We're bringing in, right there. Here's where we're delivering the hydrogens and the electrons. And you can see here's an FADH2 that's going to do the same thing. And what's left over is NAD plus, and after this, it's FAD.
Between the membrane proteins, the
See,
cell is
there it
able
goes.
to
There's the
capture
electrons
energy and use it
as
to
the
produce
hydrogens
ATP
move
molecules.
across.
Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that enter the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
Okay, so that is cellular respiration. What she said in that video is several things. At the end, she kind of tied everything together, which I'm going to do here in just a second. Let's first kind of explain how we form the ATP during this electron transport chain process. It's a kind of a cool, easy thing, and this illustration doesn't really show how kind of easy and cool it really is. But this is demonstrating what's called chemiosmosis. The actual production of ATP in cellular respiration is going to take place through this process. It involves the pumping of protons, which is, once again, just another way to describe H+ ions, okay, through the special channels in the membranes of the mitochondria from the inner to the outer compartment. Essentially, pumping it from the matrix into this space. That's what these different pumps are doing, okay? Notice they've got numbers 1, 2, 3, and 4. As the electrons move from protein to protein, what they do is they pump hydrogens into that space, okay, which leads to a high concentration of hydrogen ions here and a lower concentration here inside the matrix. Now, what do we know about gradients? Okay, we always want to go down them, right? Well, this enzyme, literally called ATP synthase, is what allows hydrogen ions to go from high to low. And they call it ATP synthase because guess what it does? It synthesizes ATP. Okay, the moving of those hydrogens through this enzyme basically just energizes it. It gives it the energy necessary to attach that phosphate to the ADP, forming ATP. And this is where most of the energy comes from, which, like I said, from this process specifically, from the electron transport chain alone, it's a total of 32 to 34, which if you add it all up, you get 36 to 38. And that's what I want to do and talk about here, the summarization of it, which is what she did in the video. Okay. For every molecule of NADH plus H plus that drops off its hydrogen atoms to the electron transport chain, 2 or 3 molecules of ATP, or an average of 2.5, are produced via oxidative phosphorylation. Okay. You might be wondering, why is it not a set number? Okay? And I'll explain that here in a second. Okay? For every molecule of FADH2 that drops off hydrogen atoms to the electron transport chain, only 1 or 2 molecules of ATP, for an average of 1.5, are produced via oxidative phosphorylation. And this is because of the fact that FADH2 drops off its hydrogen atoms at a lower energy level, complex 2, than NADH plus H plus, which is complex 1. Okay. You didn't notice in the video, and it showed it, and it does show it here as well. This electron and hydrogen carrying vehicle is bringing those electrons and hydrogens to complex 1, this one right here. Okay. Whereas FADH2, it comes farther down the line at the second complex. Okay. And why is this important, or why does this matter? Because when you bring it to the first stop instead of the second, you get more hydrogens into this space. Whereas if you're dropping them off later on down the line, you get less hydrogens into that space. So that's why FADH2 doesn't provide us with as much ATP as NADH plus H plus. Okay. But when you add it all together, one glucose molecule results in the formation of 36 to 38 ATP. And it's through those 4 steps, the net of 2 from glycolysis, we can see right here, taking place in the cytoplasm. We don't form any during the transition reaction, which is what I'm circling with my mouse right now. 2 more from the Krebs cycle. So between those 3 steps, we've got 4. And then from the electron transport chain, it's 32 to 34, which if you add 4 to 32, that's 36. If you add 4 to 34, that's 38. So this is the total sum. Total sum. My goodness. All those 4 steps: glycolysis, transition reaction, Krebs cycle, electron transport chain. Okay. That's what we call aerobic respiration. Okay. Using oxygen, producing ATP using those 4 mentioned pathways. Now, the summary of those 4 stages of cell respiration also not only includes glucose, but also needs to have vitamins and minerals. That's why we need to have a very well-balanced diet. Okay? Because NAD and FAD, as we learned earlier, are made up of vitamins. Okay? Those different proteins of the electron transport chain involve vitamins. Okay? So on and so forth. Okay, so eating not only provides us with a potential energy source, but it also gives us vitamins and minerals. To release that energy from food, though, we must breathe. The oxygen we inhale is essential for the completion of cell respiration. The CO2 we produce is exhaled in order to help keep our pH in balance. Okay. Now, this is a nice illustration showing you those 4 steps, the total number coming from them. Here's just a table you'll find in your textbook summarizing it and kind of breaking it down step by step. Do whatever you want to study. You know, the main thing is know the total for the entire process, 36 to 38. Or another illustration here, not the best one in the world, but it does show each of the 4 steps. Glycolysis, there's your net of 2, transition reaction, Krebs cycle, and then the big payoff, which is through the electron transport chain. Now that was the oxidation of glucose, or the breakdown of glucose, going from big to small, which is a form of catabolism. That's not the only type of metabolism, though, that we have. And it's not the only way to metabolize a carbohydrate. We can also build stuff up, and that would be called carbohydrate anabolism. So even though most of the glucose in our body is catabolized to generate ATP, glucose may take part in or be formed via several anabolic reactions. For example, if glucose is not necessarily needed right away for ATP production, what we can do is combine it with other glucose molecules forming glycogen, which is just the polysaccharide form of glucose, and is going to be formed in a process called glycogenesis. And I mentioned that earlier in the lecture. We also talked about this stuff back when we talked about the endocrine system. And we learned, for example, at that time that glycogenesis is stimulated by the hormone insulin whenever we are hyperglycemic, which is a fancy way to describe high blood sugar. Where we store glycogen is kind of two main places. Our liver cells called hepatocytes and in our skeletal muscle. We do that so that if later on we need to make a lot of ATP, like during exercise, for example, that glycogen stored in our liver and our muscle cells can be broken down into glucose to be released into the blood or for uptake by our cells for cell respiration. And that splitting of glycogen into glucose is known as glycogenolysis, stimulated by hormones glucagon and epinephrine in response to hypoglycemia, which is the opposite of hyper. It's low blood sugar. Remember, I told you guys to remember glucagon is released when glucose is gone, right? Epinephrine, we also have described that several times this semester as adrenaline, right? If you're during a fight or flight response, don't you think we need to make a lot of energy? Absolutely. Okay. We can also make glucose in cases where we don't have sufficient glucose in our body from non-carbohydrate sources, from things like triglycerides or fats and proteins. Like if we are low in liver glycogen or in a fasting state. The glycerol part of a triglyceride is a 3-carbon molecule, which is half as many as a glucose molecule would have, which is 6. We can use lactic acid, which is not too far off, as we've already talked a little bit about, from pyruvic acid, which is half of a glucose. And certain amino acids can be converted in our liver to glucose. And in doing this formation of glucose from non-carbohydrate sources is another one of those big buzzwords that I previously mentioned, which is gluconeogenesis. And that's stimulated by the hormones cortisol, which, as we learned previously, is one of those hormones with a nickname. It's our stress hormone. It's a glucocorticoid released by the adrenal cortex. The adrenal cortex, remember, is the outside of our adrenal glands. Glucagon also stimulates this process, and so does the T3, T4 thyroid hormones. So what I'm going to do, and I've already done this for you guys, but it's not a bad idea to kind of refresh your brains on these things. Let's just quickly type up what each of those processes is. If you forgot, this will help you tremendously. Starting with glycolysis, which is the breakdown of glucose, and to form ATP. That was what we talked about earlier today. Spelled similarly and sounds similar to it is glycogenolysis, which is the breakdown of glycogen, not glucose, and the breakdown of glycogen to form glucose molecules. That's what we do, for example, if our blood sugar is low. And that's what, for example, glucagon will trigger. The opposite of that is not glycogenesis, formation of glycogen from glucose molecules. That's what insulin stimulates. And then you got gluconeogenesis, which is the formation of glucose molecules from non-carbohydrate sources. So like fats, proteins, lactic acid. Hopefully those words ring a bell to you. while I got my Word document up here, I'm also going to add two other words that we've seen before. I mentioned this one earlier today. Lipogenesis is the formation of fats or triglycerides. And then lipogenolysis, or excuse me, not lipogenolysis, lipolysis, tried to combine a couple words there, is the breakdown of fats or triglycerides, which can be done just like with glycolysis to form ATP. And we'll get into talking a little bit more about those here in just a second. Hopefully all that rings a bell. We've talked about it all before. That does not ring a bell. You can go back in time and look at your endocrine lecture, and hopefully you wrote those 6 things down earlier this semester. If you didn't learn them at that time, or if you didn't remember them, now you hopefully will. On this page alone, we see glycogenesis, we see glycogenolysis, we see gluconeogenesis. The only one of the carbohydrate related ones not visible here is glycolysis, and we talked about that earlier in the lecture. Okay. So those first 4 I typed up are all related to carbohydrates and are all a form of metabolism associated with carbohydrates, whether it be building up like in glycogenesis or breaking down like in glycogenolysis. A couple illustrations here on the next page. On the left side, we show, and it's just kind of an easy way to demonstrate how we can go from glucose to glycogen. Looking with the red arrows, we go in this case through the process known as glycogenesis. Whereas if you go from glycogen back to glucose, we'd call that glycogenolysis. Okay. Whereas over here, the process of gluconeogenesis, remember, is the formation of glucose from non-carbohydrate sources. So from things like lactic acid, amino acids, which we know are in proteins, glycerol, which is in triglycerides, or even pruvic acid back to glucose. So formation of new glucose that way. Not that you need to worry about all these different steps. It's very complicated, as you can see, but I included it because it's in your textbook and demonstrates 3 out of those 4 big buzzwords. Oops. We'll talk about proteins now as a energy substrate. Because remember, and I talked about this in our last lecture, there's 3 major fuel nutrients: carbs, lipids, and proteins. Proteins, honestly, though, are not a typical source of energy. Okay? We don't prefer to make energy from proteins. And as we've learned before, proteins are made up of smaller molecules known as amino acids. Primary use for those amino acids we attain from food is not for energy, but is the synthesis of new proteins. Whether that's actin and myosin in your muscles, transport protein in your cells, hemoglobin in our blood, an enzyme, whatever. We talked about all the different functions of proteins and how important they are in the body at several points this semester. Okay. When we have an excess amount of amino acids, which means that they're not necessarily needed immediately for protein synthesis, we may use them for energy production. But first we have to deaminate them. Deamination means to remove this part of it, what's called the amino group. Okay. The NH2 from it Okay, and this occurs in the liver. And what is left over as a result of this process can then be converted to a molecule that will fit into the Krebs cycle. I've got to figure out this right here, essentially. Okay. Certain set of amino acids can be converted to acetyl-CoA, which then can enter the Krebs cycle, just like if you had started with glucose. Okay. Some amino acids can be converted to pyruvic acid before being converted to acetyl-CoA. Sometimes amino acids can directly go into the Krebs cycle. Sometimes they have to be directly converted to acetyl-CoA. But regardless, once we form that acetyl-CoA, then we can go to the Krebs cycle and get to the electron transport chain, which therefore allows us to produce energy. Okay. But a side effect of performing this deamination is if that amino group enters the blood, it forms into ammonia, which is NH3, which is very toxic to your body. Okay. Fortunately, though, our liver is going to convert that ammonia to urea, which is much less toxic and is eliminated in our urine. And we'll talk about urea in our next lecture when we talk about formation of urine. Like I said, we typically don't prefer to make energy from proteins. It is possible, and it's something that we do utilize from time to time, but we prefer carbs and fats. So lipids, like carbohydrates, can be catabolized to produce ATP. In fact, we can get a lot of energy from fats, a lot of energy. Okay. If our body has no immediate need to use lipids in this way, though, we store them as triglycerides in our adipose tissue. And that's especially in our liver and other parts of the body. Unfortunately, the typical American diet and the typical American lifestyle, where there's not enough exercise, results in the formation of triglycerides in our body. That's what lipogenesis is. Okay? And it's not necessarily always forming fats by eating fats, because we could convert carbohydrates that we don't utilize. So if you eat a lot of pasta and you don't exercise, you're bringing in a lot of glucose, you might end up getting fat because you can convert the glucose into triglycerides. Okay. When triglycerides are, though, to be used for energy, we use lipase enzymes to break them into their building blocks. Remember, a triglyceride is made up of kind of four things. You got a singular glycerol molecule, which is a 3-carbon molecule, and then 1, 2, 3 of these fatty acid molecules. Okay. That's the process called lipolysis, the breakdown of fat. Okay. Now, glycerol is a 3-carbon molecule, very similar to pyruvic acid. And if I jump ahead, fatty acids, 3-carbon molecule, just like pyruvic acid, you slap a coenzyme A to it, you've essentially got acetyl-CoA. Therefore, you can jump into the Krebs cycle. Okay. But, sorry, I was showing you the wrong thing. Here's where I needed to be, right here. Glycerol, 3-carbon molecule, like pyruvic acid, then you can get to acetyl-CoA. And what we can get a lot more energy from, though, is through these guys, the fatty acids. And look at the number of carbons you see in each of those fatty acids. That's where the energy's at. Okay. What we do to those fatty acids is a process called beta oxidation, where we take those long carbon chains, sometimes can be 20 carbons in length, and we break them down into 2-carbon segments. Okay. A 2-carbon segment, then you slap a coenzyme A to it, gives you acetyl-CoA. Now you might have to do a little bit of converting, but essentially you're creating the same thing that you did during the transition reaction when you started with pyruvic acid, and that's the formation of acetyl-CoA. But it's not just one acetyl-CoA, because if each of these is 20 carbons in length, that means for every one of these little fatty acid tails, you can get 10 acetyl-CoA molecules, which means that's per fatty acid tail or fatty acid molecule, that's 10 different things entering the Krebs cycle. And look at the fact that you got 3 of these. So that's like 30 molecules of acetyl-CoA entering the Krebs cycle, which explains why you can get around 100 ATP from this singular triglyceride. And that explains why, if you ever wondered, you might not have ever wondered this, things that have a lot of fat in them are higher in calories. And you get a lot more energy. That's what calories are. They tell you, a calorie tells you how much energy you get from that food source. Fats are loaded with lots and lots of energy. Make sense? Now, have you guys ever heard of ketones? What about the keto diets? That's a pretty popular diet anymore. As part of normal fatty acid catabolism, as the normal breakdown of fatty acids occurs, our liver converts some of those acetyl-CoA molecules into substances known as ketone bodies, or just ketones. Okay, this includes acetoacetic acid, beta-hydroxybutyric acid, acetone. You don't need to know those fancy names, but those are some examples of ketone bodies. Formation of these substances is called ketogenesis. Genesis, once again, creation, and so we're creating ketones. Okay. Once they're produced, these ketone bodies enter or leave the liver, where they enter our body cells, and there they can be broken down into acetyl-CoA to enter the Krebs cycle. The level of ketone bodies in our blood normally is very low because other tissues use them for ATP production as fast as they're formed. But during periods of excessive beta oxidation, production of them exceeds their uptake and use by our body cells. And this occurs like after you eat a very rich, triglyceride-rich meal, or if you're like in a fasting or starvation state, because few carbohydrates are available for catabolism. So if you don't have carbs to get energy, you have to make their energy some way. You're going to start making these ketones because you're going through a lot of that beta oxidation. You're using your fats. Okay. That is the whole theory on the keto diet. If you aren't eating a bunch of carbs, essentially a keto diet is kind of another way to describe a low carb diet. Okay. Every so often you hear these diets kind of just changing names. Low carb diet used to be called the Atkins diet. You may have heard of that before. It's just the same essentially thing that you've probably encountered before if you follow, diet, exercise, and that kind of stuff. Okay. You also might experience excess beta oxidation if you're a poorly controlling diabetic, okay, or you're untreated diabetic in the cases of people with diabetes mellitus. Okay. Now, when the concentration of ketone bodies in our blood rises above normal, a condition called ketosis can occur. And those ketone bodies, most of which are acids, must be buffered. If too many ketones are accumulating, our blood pH falls, and that causes ketoacidosis. Decreased blood pH in turn causes depression of the central nervous system, resulting in disorientation, coma, and even death if the condition is not treated. And that can happen in, like I said, uncontrolled or untreated diabetes, and can be deadly. Okay, obviously, if death is involved, it's deadly. When a diabetic becomes severely insulin deficient, one of the telltale signs is the sweet smell on their breath from the ketone body acetone. They describe the breath of these diabetics as being like really fruity. Anybody ever heard of this before? They not even their breath sometimes will smell fruity. They say their urine will as well. And that's kind of weird, but that's what actually happens. So if you've ever heard that, now you know why. So that's our summarization of metabolism. We started today talking about catabolism of glucose, making a bunch of ATP through glycolysis, Krebs cycle, electron transport chain. Talked a little bit about anabolism of glucose, building glucose or building up glycogen from glucose. We talked just now about catabolism. I just briefly mentioned anabolism, didn't get too much into that, but I did define what lipogenesis is. We talked before that about deamination, taking amino acids and getting energy from them through protein catabolism. What I did not mention anything about in this lecture is the anabolism, the building up of proteins. And that's because we've already covered it. Way back at the beginning of the semester when we talked about DNA, RNA, we talked about ribosomes, the endoplasmic reticulum, gene expression going from DNA to RNA to proteins. That's what this is. So we've already talked about it. Taking DNA, making RNA from it, and then taking amino acids and putting them together, using our ribosomes, the organelles that we call protein factories because they are the ones that make the proteins. So I didn't really need to do that again. The last thing I want to talk about is utilizing our metabolism to give us our body temperature. Producing heat, not necessarily as a bad thing, but as a beneficial thing to give us our 37 degrees Celsius or 98.6 degrees Fahrenheit body temperature. So there's a nice relationship between your metabolic rate, your metabolism, and the heat that you produce. Okay? Your body's going to produce more or less heat depending on the rate of its metabolic reactions. Okay? The higher your metabolic rate is, the higher your body heat production is. Okay? You produce more heat when you are going through more metabolism. Okay? And because homeostasis of body temperature can be maintained only if the rate of heat loss from the body equals the rate of heat production by metabolism, it's important to understand the ways in which heat can be lost, gained, or conserved. Heat, we've previously talked about this, is a form of it's a form of kinetic energy. Okay, it can be measured as temperature. Despite wide fluctuations in environmental temperature, homeostatic mechanisms can maintain a normal range of internal body temperature. And we've talked about things like shivering and sweating before. Okay? It's quite a bit more complex, though, than just shivering and sweating. Okay? If the rate of body heat production equals the rate of heat loss, so in other words, if we produce as much heat as we're giving off, body maintains a constant core temperature, which is what we want, around 37 degrees Celsius or 98.6 degrees Fahrenheit. Since heat is a byproduct of metabolism, it is important to understand metabolism to understand body temperature regulation. Okay. I've mentioned this before, but your basal metabolic rate, or BMR, is a measurement of an individual's metabolic rate with the body in a quiet, resting, and fasting condition. So the best way to measure your BMR is right when you wake up after sleeping like 9 hours or so. Okay. Before you've gotten out of bed, even started walking around, because as you guys know, contracting your skeletal muscles is going to produce heat. The energy that your muscles are using produces heat. Even before you've drank or eaten anything, because that activates digestive mechanisms, which are all chemical reactions that produce heat. And that's why fasting's key. Okay? Now, there's easy ways to measure your body temperature, like, you walk into businesses now, especially after the pandemic, you can get your face scan. They have the thermometers that you can swipe across your forehead. When I was a kid, I remember sticking a thermometer underneath my tongue, or when you're a baby, they, do the armpit or, into the anal canal. What you're measuring in that case is what you would call your shell temperature. Okay, the temperature near your body surface. In other words, in your skin or subcutaneous layer, which is not always indicative of what your true internal core temperature is, which is the temperature in your body structures deep to our skin and subcutaneous layer. That's the temperature that's usually around 37 degrees Celsius. And if that core temperature is too high, it will kill our cell, or it will kill you, because it denatures your body's proteins. Proteins start unwinding, and then they start losing function. Whereas if you have a core temperature that's too low, that can cause your heart to have a abnormal rhythm, cardiac arrhythmia, which can trigger death. Okay. Now, depending on the environmental temperature, your cell temperature is usually 1 to 6 degrees Celsius lower than core temperature. It can also be higher. Like if you're sitting in a sauna, or if you've been outside in the heat, your cell temperature is probably going to be higher than your core temperature. Okay. As a tissue increases its metabolism, the amount of heat it produces also increases. Several factors influence your metabolic rate, which therefore affects your rate of heat production. If you increase your metabolic rate, you increase your heat production, therefore core temperature will go up. Okay. Things that affect your metabolism, therefore affect your heat production, include exercise, the release of the thyroid hormones, epinephrine and norepinephrine release from our sympathetic nervous system. Your increased body temperature actually affects your metabolic rate, which affects your body temperature or your heat production, which is kind of crazy. Ingestion of food, your age, your gender, the climate you're living in or you're exposed to at the time, your sleep state, and if you are in a malnourished state as well. In order to maintain our normal body temperature, we need to be able to lose heat to the environment at the same rate as the heat that we are producing through metabolic reactions. We've talked a lot about gradients this semester, whether it be concentration gradients, electrical gradients, pressure gradients. Well, we have one more gradient to talk about, and that's temperature gradients. And it's the same principle essentially we've talked about all semester, where in this case heat is going to go from high temperature to low temperature, from high to low. Okay, you've probably heard, whether it was you saying it as a parent or your parent saying it, shut the door, you're letting all the cold air in, or you're letting all the heat out. Well, which is it? Are you letting cold air in or are you letting heat out? Okay. Well, if, for example, it's the summertime and it's hot and you have your air conditioning on in the inside of your house and you keep your house at 70 degrees roughly, that's a pretty standard temperature in the house, and it's 100 degrees outside, it's hotter outside than it is inside, right? So the heat would actually be coming into your house because it's going from high to low. Whereas like in the wintertime and it's super cold outside and you're, got the door open, you'd be letting the heat out of your house from where it's warmer to where it's colder. So next time you yell at your kids, or next time your parent yells at you when you're visiting them, if you don't live with them, when they yell at you for this, make sure they yell it correctly or you yell it correctly. Okay, heat can be transferred to the environment in 4 ways: conduction, convection, radiation, evaporation. And there's examples of them illustrated on the right. Conduction is the heat exchange occurring between molecules of 2 materials, and the key here is that they're in direct contact with each other. Okay, like when you walk into a classroom, like the one I have here in Atchison, with the room temperature fairly cold, so too is the table that you sit at. So too are the chairs that you sit in. And if you put your arm onto that table, you can feel that coldness. And what's going to happen is your body is going to lose heat to that table. The table is going to warm up because you're transferring your heat to it. Okay. Another example of conduction happens if you sit in a hot tub. That's where you would actually probably gain heat because the hot tub is maintaining a temperature higher than your body. Okay. Now, sitting in a hot tub also is a form of convection. And convection is the transfer of heat, but not with molecules in direct contact with one another, but with fluids, which in the case of this is actually can be a gas or a liquid. Okay. Not just a fluid, what you would think of as a liquid like water, but also gas, like the air around us. Okay. Like when you fan your face, that helps cool your body because you're performing convection. Okay. Now, what do you think? Are you able to transfer heat to the air or to water more effectively? What do you think? Air? What do you think? Water. It's water. Okay. Have you ever been outside on a 70-degree day? It feels fantastic. Sunday was a perfect day. It was like 70 degrees. You ever been swimming in a pool that's 70 degrees? That's cold as, you know what? That feels awful. Okay. It doesn't feel pleasant to be in a 70-degree pool. Okay. That's because the water, like, it snatches your heat a lot better than the air does. Okay. That's why if you fall into water that's like 50 degrees, you're in danger. Whereas like if you're outside without a jacket on a 50-degree day, you might be a little bit cold, but you're not in danger. Okay. Radiation is transfer of heat in the form of infrared rays between a warmer object and a cooler one without physical contact. That would be like, for example, how we get our heat from the sun. And then evaporation is the conversion of a liquid to a vapor involving both evaporated water from the skin, but more important, which is way more important than when we exhale water from our lungs. Okay. The best way to cool yourself off when you are exercising, unless you're exercising in a pool, is to allow your sweat to evaporate. That's why like Under Armour and like Nike Dry Fit, those are so great, much better than like a cotton t-shirt, which just gets soaked. That wicking away of that moisture, that sweat, makes it evaporate more easily, and that pulls the heat off your body much more efficiently. All right, last slide here. A control center, as we've learned previously, functioning as your body's thermostat is up in your hypothalamus in its preoptic area. It receives impulses from the thermoreceptors in our skin, as well as the mucous membranes, and even within the hypothalamus itself. Okay. As a result of that input, the hypothalamus carries out signals that are either going to promote heat or to promote heat loss. Okay. I shouldn't say promote heat formation. In other words, trying to raise body temperature. We know when we get cold, what do? We shiver. But not only do we do that, we constrict the blood vessels in our skin in order to keep that heat in our blood closer to our core and less close to the skin, so less heat will be lost to the environment. We also would redistribute the blood to the more important internal organs, which therefore might sacrifice a finger or a toe, but you can survive just fine without one of those. You can't survive too well without one of your internal organs. Okay. On top of that, we're also going to be releasing the thyroid hormones epinephrine and norepinephrine because of their role in increasing metabolism. Because if you increase your metabolism, that increases your heat production. Okay. So aside from shivering, we do two other very important things when we get cold. And then when we get hot, what do? We sweat. And that's the most obvious thing, and we've talked about that this semester. But we also do things to decrease our metabolic rate, which decreases our heat production. And then we dilate the blood vessels in our skin, which means that the blood will be pooling in our skin and thus more likely to dissipate the heat to the environment, whether that's through the sweat or like fanning your face and performing conduction. All right, any questions?